The UA gene, described here, expresses a protein involved in a pathway to stimulate feelings of alertness in the presence of food. A gene contains a core promoter element and is regulated by a distal enhancer called EE1, which is 3,000 base pairs upstream of the core promoter element. The EE1 also possesses a silencer element, which is within its sequence and functions to inhibit transcription when bound to the transcription factor full in a sequence-specific manner.
First general transcription factor to bind are the TATA binding proteins, which bind to the TATA box in a sequence-specific manner and serve as a platform for TF2D and TAF binding. The TAFs do not interact in a sequence-specific manner, but can interact with the motif 10 element, downstream promoter element. After TF2D binding, TF2A binds to the TF2D complex and stabilizes its interaction with DNA, and subsequently recruits transcription factor 2B, which binds in both the B recognition element upstream and B recognition element downstream to further stabilize the complex's interaction with DNA. Transcription factor 2F binds to RNA polymerase 2 and recruits it to the pre-initiation complex. Transcription factor 2F subsequently recruits transcription factor 2E, and transcription factor 2E recruits transcription factor 2H. Transcription factor 2E is necessary for promoter melting. Transcription factor 2H has helicase and kinase activity, the helicase needed for the open complex formation, and the kinase activity is required as it recognizes the repeat heptad on the C-terminal domain of RNA polymerase 2. When in the presence of food, olfactory stimulations from the food cause the creation and release of the HNG hormone. The HNG binds to the hunger response element on the extracellular face of cell membranes, and upon binding causes the dissociation of the HRE complex and movement, translocation into the nucleus, and binding at the enhancer element 1.
HNG protein binds to the mediator complex, which is bound to the promoter initiator complex.
Transcription factor 2H and transcription factor 2E help to stimulate the melting of the DNA at the +1 site to form the open promoter complex. That open complex formation has been achieved. Nucleotide triphosphates begin to move into the active site channel of RNA polymerase 2, where they begin to deposit onto the template strand. Initially, the RNA polymerase 2 can experience abortive transcription, where nucleotide synthesis is not efficient enough, and the DNA scrunches, and the transcript is kicked out of the active site channel. Once the RNA polymerase 2 has synthesized approximately 10 nucleotide length of RNA, the RNA begins to move out of the exit channel. The incorporation of the 10 nucleotides favors promoter escape over abortive initiation. After 25 nucleotides have been synthesized, mediator places the C-terminal domain of the RNA polymerase 2 near transcription factor 2H, which has a repeat heptad that is recognized by the kinase functioning of transcription factor 2H, which phosphorylates it.
So after 25 nucleotides have been transcribed, the capping enzyme will recognize the phosphorylated CTD element, and that will form a 5' cap on the newly formed RNA strand, and that begins the elongation complex.
So while the HNG-HRE complex are stimulated by the olfactory secretions, there's also the full protein, which binds to the silencer element, which will inactivate and cause the formation of repressive chromatin structure by outcompeting the HNG-HRE for the silencer element. HRE complex upregulates transcription of the AWA gene by binding to the distal enhancer element, while the full protein, which is stimulated after food is consumed, binds to the silencer element and outcompetes HNG-HRE and forces downregulation of the AWA gene.
The distal enhancer element, we also have a silencer element, which binds the full protein, outcompeting the H&G-HRE complex for binding in a sequence-specific manner. Full protein downregulates transcription by promoting repressive chromatin structure downstream of the distal regulatory element, including the AWA gene, preventing the formation of the transcription initiation complex and preventing transcription.